Today we're going to talk about something pretty incredible. It's a medical imaging technique that lets doctors basically journey through the tiny invisible highways of blood vessels at the back of your eye. It's all about mapping the unseen to catch problems a normal photo could never ever find. Okay, let's get right into it.
So just try to picture this for a second. Deep inside your eye, there's this delicate web of blood vessels called the retina. It's thinner than a piece of paper. Now, imagine there's a microscopic leak somewhere in that network. How on earth do you find it? Seriously, it's like trying to find one specific leaky pipe in an entire city's plumbing system, all at once.
You see, a standard picture of your retina is, well, it's just a snapshot. It's a still photo. And while it's useful, it can't show the one thing that really matters here: movement. It doesn't show you where the blood is actually flowing, where it might be getting stuck, or most importantly, where it's leaking out. To find that leak, doctors have to see the whole system in motion.
And that is where this ingenious solution comes in. It's a test called Fundus Fluorescein Angiography, or FFA for short. Basically, it's a clever way to use a special fluorescent dye and a super fast camera to create what's essentially a live-action movie of the blood flowing through your eye. So how does this actually work? Well, it all comes down to a really clever trick of physics that involves a very specific dye and two different colors of light.
And that trick is called fluorescence. It's this really cool property that some molecules have, where if you hit them with one color of light, they absorb all that energy, and then, almost instantly, they glow back a completely different color. You can think of it like setting off a tiny light-activated glow stick inside your bloodstream.
And here is where the magic happens. It's all about two perfectly matched filters. Take a look at this diagram. First, a flash of pure blue light goes into the eye. That's the exciter light. The special dye that's now in the blood vessels absorbs that blue light, and immediately, as you can see on the graph, it emits a new yellow-green light. Then, and this is the key, a second filter, the barrier filter, blocks out all of that original blue light and only lets that brand new yellow-green glow reach the camera.
So, it's this two-filter system that makes it all work. Let's break it down. Step 1, a blue light flashes to excite the dye. Step 2, the dye flowing through your blood vessels absorbs that light and starts to glow. And Step 3, the camera, which is sitting behind that barrier filter, only captures the glow. What you get is this incredible high-contrast image where the blood vessels just light up like neon signs against a perfectly dark background.
Okay, so now that we've got the science down, let's follow the dye on its ridiculously fast journey. We're talking about a trip from a vein in your arm all the way to the microscopic capillaries in the back of your eye. It is truly a race against the clock.
So how does this race begin? It starts with a pretty simple step, actually. A small amount of this water-soluble dye, called sodium fluorescein, is injected into a vein, usually right in the patient's arm. And that's the starting pistol for the whole show.
And get this, it only takes about 10 seconds. 10 seconds for that dye to travel from your arm, race through your heart, shoot up to your head, and light up those incredibly tiny vessels in your eye. In the time it takes to take a deep breath, the show is ready to begin.
And this timeline here shows you exactly what doctors are watching for second by second. First, at around 10 seconds, they see a faint, kind of patchy glow. That's the deep layer called the choroid filling up. Just 2 seconds later, boom, the retinal arteries flash bright white. By 30 seconds, the veins are full and the whole retina is at its peak brightness. But some of the most important clues actually show up much later, around the 10-minute mark. As the dye starts to wash out, any abnormal areas that are leaking or staining really start to stand out. All right, so the doctor now has this whole series of glowing images, taken over about 10 minutes. But what are they actually looking for? How do you translate these beautiful, abstract patterns of light and dark into a real diagnosis?
Well, it really all boils down to two very simple ideas: hyperfluorescence, which just means an area is way too bright, and hypofluorescence, which means an area is way too dark. Believe it or not, pretty much every diagnosis that comes from an FFA is based on finding and understanding these two simple patterns.
So let's talk about too bright, hyperfluorescence. There are 4 main reasons this can happen. First, you've got leakage. The blood vessel's watertight seal is broken and dye is just spilling out. Then there's pooling, where the dye collects in a very specific pocket, kind of like a blister. Staining happens when some abnormal tissue, like a scar, actually soaks up the dye and holds onto it. And finally, you can have what's called a window defect, which is just when a layer of pigment cells is missing, basically opening a window and unmasking the normal glow from the layer underneath. And on the flip side, an area can be too dark, for two main reasons. The first is a simple blockage. Something, maybe a bit of blood, is physically in the way, blocking the camera's view of the glow. But the second, a filling defect, is more like a traffic jam inside the vessel. The road is blocked, so the dye-filled blood can't even get to that area in the first place.
So by piecing together these clues, these patterns of light and dark, doctors can diagnose a whole range of eye conditions with incredible precision. Let's look at a couple of real-world examples of how this all plays out.
Take a patient with diabetes. An FFA can be just amazingly revealing. These tiny bright dots of hyperfluorescence, those are the earliest signs of weak spots in the vessel walls. These dark patches of hypofluorescence, those tell the doctor that parts of the retina are literally being starved of blood and oxygen. And these intense, fluffy-looking areas of leakage, that's the telltale sign of fragile, dangerous new blood vessels growing where they absolutely shouldn't be.
Or how about this condition, CSC, where a blister of fluid forms under the retina? An FFA might show a classic ink blot pattern or sometimes a smokestack of leakage. Now, this doesn't just confirm the diagnosis, it pinpoints the exact source of the leak, giving the doctor a precise target they can potentially seal up with a laser. It's incredible.
Okay, a procedure that involves injecting a dye directly into your bloodstream. That sounds a little intense, right? It's a totally fair question to ask, is it safe?
Well, the data is pretty clear. While some minor side effects can happen, serious ones are very, very rare. About 5 to 10% of people might feel a quick wave of nausea that passes, but a severe allergic reaction, anaphylaxis, happens in less than 0.05% of cases. Medical teams are always prepared for it, just in case, but it is exceedingly uncommon.
And all of this is why, even with all the amazing new imaging technologies we have today, this quote really holds true. When it comes to finding active, real-time leaks and guiding treatments that can literally save a person's sight, FFA is still the absolute, indispensable gold standard in clinics all around the world. When you really stop and think about it, what this technique does is fundamentally profound. It makes the invisible visible. It takes a dynamic, hidden process happening deep inside our bodies and turns it into a clear, actionable map. And it really leaves you wondering, what other secret workings of our biology are just waiting for us to find the right trick and the right kind of light?